The closure of the neural tube does not happen at the same time along the entire anterior-posterior axis. Recall that in the chick, which is not shown here, the posterior portion of the embryo is still undergoing gastrulation as neurulation is happening anteriorly. And the closure of the neural tube begins at the level of the midbrain in the chick embryo. In mammals, there are multiple points at which the neural tube closure is initiated. So in mice, as you can see, there are 3 closure points. And in humans, 5 different closure points have been described, as you can see here. So the closure of the neural tube begins at these points and then it extends anteriorly and posteriorly from each point. As I mentioned near the beginning of this lecture, the portions of the neural tube that close last are the most anterior and the most posterior regions. And these 2 open ends are known as the anterior neuropore and the posterior neuropore. And you can see them in this developing human embryo. Take a look at the following videos to see what the process of neural tube closure looks like in the mouse embryo. While the exact mechanism behind the closure of the neural tube is not completely understood, the cells of the epidermal ectoderm surrounding the closed neural tube have been shown to extend filopodia, which is what you're seeing here, toward each other. However, the function of these filopodia is not currently known. What is known is that different cadherins need to be expressed in the neural tube and the epidermal ectoderm in order to allow for the proper closure of the neural tube and the separation of these 2 tissues. So initially, the ectoderm expresses E cadherin throughout, but when the neural plate forms, these cells stop expressing E cadherin and they begin to express N cadherin instead. That change in cadherin expression in the neural plate promotes the separation of these tissues, since cadherins, remember that they bind preferentially to the same cadherin type. When N-cadherin mRNA is injected into the cells of the epidermal ectoderm, which express E-cadherin, what you see is that the neural tube doesn't close properly. So the presence of N-cadherin here prevents the separation of that epidermal tissue from the neural tissue. Well, knocking out N-cadherin prevents the formation of the neural tube, and that suggests that defects or mutations that affect cadherin expression can lead to neural tube defects. As you can see from this picture of a developing human embryo, the process of neurulation happens very early in development. In humans, this process is happening between days 22 and 23 of embryonic development, and the anterior and posterior neuropore close at around day 27. Now, the fact that this happens so early in development is perhaps one of the reasons why neural tube defects are still quite common. They occur in about 1 of every 1,000 live births. A woman may not realize that she's pregnant until after this point, and there are certain factors that can contribute to neural tube defects. We're going to discuss the factors that can contribute to neural tube defects in a moment, but first let's look at what these defects are. If the posterior neuropore fails to close, a child will be afflicted with spina bifida. The severity of spina bifida depends on how much of the spinal cord remains exposed. So in some cases, there are no symptoms that are apparent, while in more severe cases, spina bifida can result in things like poor bladder control and difficulty walking. Now, failure to close at sites 2 and 3, which are located, as you can see here, in the rostral or cephalic region, lead to anencephaly. And in that condition, the forebrain, which is the largest portion of the brain, remains exposed, and the amniotic fluid causes it to degenerate. Now, you can see in this picture a child, her name is Victoria, who had anencephaly, and she lived to 2 years of age. But that's very rare. The vast majority of children who are born with anencephaly don't survive more than just a few hours or a few days. There are both genetic and environmental factors that can contribute to neural tube defects. For example, the mouse that you see in this picture has a mutation in a transcription factor that's known as grainy head-like 3. And that transcription factor is required to downregulate E-cadherin in the neural folds. So in the absence of the transcription factor, E-cadherin in the neural folds is not downregulated and the neural tube does not close properly. As you can see, this mouse embryo has spina bifida, and it also has a condition known as midbrain exencephaly. This is where the brain tissue is outside of the skull, and that happens before the tissue degenerates, leading to anencephaly. The environmental causes that are associated with increased risk of neural tube defects include things like diabetes, obesity, toxins, drugs, and diet. Now, one of the most common causes of neural tube defects is folic acid or folate deficiency. And that's the reason why folic acid should be taken by women that are of childbearing age, and also why certain foods like cereals and grain products are fortified with folic acid. Now, the underlying role of folic acid is not fully understood, so the reason or reasons why folic acid deficiency causes neural tube defects are not known. It's thought that folic acid may play a role in DNA methylation or histone modification because it is used for the synthesis of S-adenosylmethionine, which is a methylation agent that is used for DNA and also for histone methylation. The epigenetic changes that are caused by folic acid deficiency then are thought to promote these neural tube defects. This hypothesis is supported by the fact that studies have shown that alterations to demethylases can cause neural tube defects. Folic acid is also important for regulating DNA synthesis, particularly in the brain. So it's possible that folic acid deficiency results in decreased cell proliferation in the area of the neural tube, which is needed to promote the closure of the neural tube. Another risk factor for neural tube defects is zinc deficiency. In a study that was carried out in 2018, it was found that zinc deficiency leads to the stabilization of the transcription factor P53, and this transcription factor promotes apoptosis. So what was found is that when mice were treated with a zinc chelator, which is an agent that binds tightly to zinc ions, so these mice were zinc depleted, the levels of apoptosis increase. And those are shown here, apoptotic cells are shown here in green and red. As you can see, the levels of apoptotic cells are much higher in the zinc depleted embryo compared to the control. While it is estimated that about a quarter of neural tube defects can be prevented with folic acid supplementation, these defects still happen and they can be life-threatening or cause disability. There have been recent advances that have been made in the surgical treatment of spina bifida prior to birth, and these advances have the potential to improve the quality of life of those affected.